وإن شاء الله يا ربي يعني تكون هاي بداية يعني يعني لمجموعة ويبينارات نحكي فيها عن مواضيع لها علاقة بالكريستي. احنا يمكن مقصرين شوي في هذا الجانب، فهلا صار يعني بدنا ندخل اكثر في هاي المواضيع. قبل ما نبدا خليني بعد ما ارحب فيكم احكي شوي صغيره عن اجون، واعمل مقدمه صغيره عن ازيرتا شركائنا اللي بحب اشكرهم كثير. هم مش فاهمين هلا، بدنا رح اشكرهم بالانجليزي بعدين. هم دائما سبورتنج لنا بكل الايفنتس اللي نحن بنعملها. هذا ثالث ويبينار بنعمله بفتره الحظر تبعت الكوفيد 19، وكانوا معنا فيه ثالث ويبينار معاهم هم. دكتوره مي اهلا وسهلا. هلا بدي احكي اول شيء عن اجون. اجون شركه اردنيه بريجنال اوتريتش. احنا بنشتغل بنعتبر حالنا انفراستراكشر للصناعه الدوائيه، بنشتغل بالصناعه بشقيها، بنشتغل مع المصانع وبنشتغل مع مستودعات الادويه. بنشتغل في عمودين رئيسيات في الشغل تبعنا. بنشتغل على مواضيع الكومبلاينس بشكل اساسي. كثير بنشتغل مشاريع كومبلاينس من بدايه انك تعمل جي ام بي سيستم من الصفر، لا انه انت تكمل ويكون عندك جابس حابب تو بريدج ذيس جابس، فاحنا بنتدخل وبنساعدك بشكل كبير بهذا الموضوع. وبنحاول قدر المستطاع نكون اب تو ديت في التغييرات المتسارعه اللي بتصير في الصناعه الدوائيه. فبالتالي بنشتغل مع المصانع لما بصير اي تعديل اي تغيير في الصناعه الدوائيه، بنحاول ان نخلي الشركات تطابق وتكون إن كومبلاينس. الجزء الثاني الفارماسيوتيكال انجينيرنج. احنا بنساعد الشركات مواضيع مختلفه ومتنوعه في موضوع الفارماسيوتيكال انجينيرنج، زي الكواليفيكيشن، الفاليديشن، الثيرمال مابينج، الفيزيبيليتي ستدي، الماركت ريسيرش. بنشتغل على هاي المواضيع بشكل اساسي وجدي. وعندنا مشاريع كبيره بنساعد شركات من تحت الانشاء بالبروجكت مانجمنت، بالمراحل اللي ما قبل حتى البروجكت مانجمنت للمشروع. اوكي. بشكل اساسي سوقنا الاكبر هو في السعوديه، بس احنا متواجدين في الاردن وبنخدم كل الشركات الاردنيه وبنخدم كل الشركات الفلسطينيه الحمد لله، وعندنا علاقات كثير ممتازه معاهم. بس لنا يعني الحصه الاكبر من شغلنا عم بتكون في السعوديه. احنا متخصصين بالسعوديه في اي جايد لاينز والجي سي سي جايد لاينز اكثر. بنشتغل بمصر. احنا فخورين كثير بالاوبريشن تبعتنا بمصر، لها حوالي 3 سنوات بس اشتغلنا فيها مع كثير شركات كبيره ومالتي ناشونال في مصر، وهذا شيء احنا كثير فخورين فيه. بنشتغل بالسودان، بنشتغل بكردستان وببغداد، وبنشتغل بالجزائر. طبعا هذا طبعا بالاضافه للاردن. بدي احكي عن ازيرتا. بالاول بدي first of all I would like to thank you everybody coming from Azierta. Thank you Marta, Sandra, Leticia, Eduardo, Pablo, Danny, everybody, Anna, because you were a very big support for us during this very hard times. You were able to support us during all these webinars. This is the third webinar that we organized together, and you have been always your webinars have been always very informative, very rich in information, very updated, and very enlightening. So it gave a very good response and a very good impression for our clients. We would like to thank you for that. It's a very tough time, but you found the time, you found the resources to support us. So thank you very much for this. I would like to talk a little bit about Azierta. Azierta is a company that was established in, it's based in Spain. Azierta, they consider themselves a global consultancy in science and health. They are aimed to science and health, and they have 4 levels of customer services: scientific, industrial, technological, and health management. They have more than 100 professional experts, and they have more than 500 clients. They have 4 divisions, as we said, in Azierta. They have different offices in Madrid, Barcelona, Paris, Lisbon, Milan, and Bogota. Azierta, for most of you joining us today, you know Azierta for their services regarding the ADE limits and the ADE reports and the PDE reports, which we have managed to service much of our clients in the region with these reports through Azierta, and they have been always up to date. They have been always state-of-the-art reports, but they offer a lot of other services, and they have very big information specifically in the subject that we are tackling today, which is the elemental impurity. The second thing I wanted to talk about is the ladies who are joining us today. I would like to welcome Marta, Sandra, and Leticia. They are gonna be taking turns on this presentation. Sandra has a master's degree in research and development, industrial production, quality and regulatory affairs of medicines. She is a toxicology technician in Azierta, and Marta has a master's degree in drug research, development, and innovation, and she is junior toxicology and quality technician as well in Azierta. As well for Leticia, she's the QA GMP responsible person in Azierta, and she has been with us in the cross-contamination webinar that has taken place like 3 weeks ago or 4 weeks ago, and it was very informative. I would like to welcome you, ladies, and I will not talk anymore because I took a lot of time. This is your webinar. Please feel free, but I would like to draw the attention of the attendees that if you have any questions, just write them in the questions and answers section, and we will answer them at the end of the webinar, at the last 30 minutes of the webinar. Go ahead, ladies. It's your territory.
Thank you, Yara. Let me share my screen.
Yeah, shared. Very good.
Okay, thank you. Okay, so good evening to everyone. First of all, we would like to welcome you to this session of evaluation of elemental impurities according to ICH Q3D guideline. The aim of this training is to acquire the basic competencies to perform risk assessments on elemental impurities in finished products following the requirements established in Q3D guideline. For this, firstly, we are going to see a theoretical basis on which we will go through every aspect of this guideline. Then we will show you some case studies to apply the acquired knowledge. Finally, as Yara said, we will answer all the questions that you may have during the training. So please take notes of everything you need to ask us, and we will be glad to answer all your questions in the last part of the training. Before starting with the training, I would like to apologize in advance for my English, so please, if you don't understand something or something is not clear, please let me know. Nevertheless, after the training, we could send you all these slides. Okay, so first of all, we need to define what's an impurity. We know as impurities all those substances that are not intentionally added to the drug product with a therapeutic benefit. These impurities may be either organic molecules, residual solvents, or inorganic material. Considering that they do not have any type of therapeutic benefit for the patient, and due to their potential toxicity, their presence in the final product should be limited. In case they are present, their levels should be guaranteed to be within acceptable limits for health. Elemental impurities are inorganic elements commonly known as heavy metals. According to ICH Q3D guideline, 24 elements are considered as elemental impurities. Later, we will talk about all of them. Elemental impurities may be intentionally added during the manufacturing process of the product as catalysts or metallic reagents, or unintentionally added due to interaction with the equipment or the auxiliary service by the interaction with the packaging material or forming part of any of the components of the product. The presence of elemental impurities in pharmaceutical products has been traditionally monitored through the so-called heavy metals test. This test is based on a sulfide precipitation reaction in which a sulfide ion-donated reagent reacts with the metallic elements contained in the sample, giving rise to a colored compound. The color intensity in the obtained solution is visually compared with a lead reference solution. It should be noted that this test only detects certain elemental impurities, assuming that all of them precipitate with the sulfide ion to the same extent. Due to the limitations it presents, currently the heavy metals test is considered obsolete. On one hand, the visual colorimetric evaluation is subjective and not very sensitive. An exact concentration of elemental impurities cannot be established, but limits between 5 and 20 ppm are usually used. In addition, the method does not allow to differentiate which specific elements are present in the product. In contrast with this method, ICH Q3D presents a fully detailed, risk-based process to assess and control the content of elemental impurities in pharmaceutical products. The heavy metals test is replaced with new analytical methods, much more sensitive and selective, such as ICP. These methods allow to assess the presence of each individual element, which gives the opportunity to manage their levels, considering the toxicological profile of each of them. The aim of ICH Q3D guideline is to limit the presence of potentially toxic elemental impurities in medicines for human use in order to ensure patient safety. It establishes a new toxicological criterion through which 24 metallic elements are classified into 3 different groups according to their toxicity and their probability of occurrence. In addition, Q3D guideline provides PDE values for each of these elements as a function of the administration route. The risk assessment is not limited to evaluating only the presence of elemental impurities intentionally added during the synthesis of the active substance. It also considers all possible sources that may contribute to the final content of elemental impurities in the product. The evaluation may be performed using 4 possible options. 3 of them are focused on the components, and 1 on the final drug product. Depending on the outcome of this evaluation, it's determined if it's necessary to establish a control strategy by comparing the estimated concentration of each element in the product against a control threshold. This guideline does not apply to the following cases, including veterinary and herbal products, radiopharmaceuticals, vaccines, or elements that are intentionally included in the drug product for therapeutic benefit among others. It does not apply neither to products used during clinical research stages of development. Let's focus now on one of the main novelties that ICH Q3D guideline provides, the classification of the 24 metallic elements according to their toxicity and their probability of occurrence. Class 1 includes elements of high toxicity by all routes. Although their intentionally use in the manufacture of pharmaceutical products is null or limited, their presence is common in materials commonly used in the pharmaceutical industry, specifically in those of mineral origin. Therefore, they need to be included always in the risk assessment, regardless of the route of administration of the product. In this category, arsenic, cadmium, mercury, and lead are included. Class 2 includes elements whose toxicity depends on the route of administration. This category is subdivided into 2 groups according to the probability of occurrence. Class 2A contain elements that have a relatively high probability of occurrence, and hence cobalt, nickel, and vanadium must be included always in the risk assessment. Class 2B elements have a lower relative natural abundance and less probability of being isolated together with other materials. Therefore, they are included in the risk assessment only if they are intentionally added. Class 3 includes elements with relatively low toxicity when administered orally. However, they must be evaluated in the risk analysis when the product is administered by parenteral and inhalation route, and when they are intentionally added. For all these elements, ICH Q3D provides PDE values for oral, parenteral, and inhalation routes. The permitted daily exposure is defined as the maximum dose of a certain substance to which an individual can be exposed every day throughout his life without suffering any adverse effect. Thus, the lower the PDE value, the greater the toxicity of the element. In this slide, You can see a summary of which elemental impurities should be considered in the risk assessment depending on the route of administration. As you can see, Class 1 and 2A elements need to be included in all routes. Class 2B elements are included only when they are intentionally added. And finally, Class 3 elements are only assessed if they are intentionally added and in parenteral and inhaled products. In parenteral products, only lithium, antimony, and copper need to be assessed, while for inhalation route, all Class 3 elements should be included. When the product is intended to be administered by a route other than oral, parenteral, or inhalation, a route-to-route extrapolation may be performed to calculate acceptable exposure limits based on existing PDE values. This extrapolation must be carried out individually and specifically for each case. Now we have described the basic principles of Q3D guideline. We are going to focus on the main steps that must be followed in the implementation process. Firstly, all the possible sources that may contribute to the final content of elemental impurities in the product need to be identified. In the guideline, 5 basic sources are given: active substance, excipients, water, container closure system, and manufacturing equipment. If any other potential source other than those mentioned in the guideline is identified, it should be considered in the risk assessment as well. Once all the potential sources are identified, each of them is analyzed. For this, it's necessary to know if some elemental impurity is intentionally added in the active substance or the excipients. In addition, all the elements potentially present, even if not intentionally added, need to be identified for all sources. Later, we will see which documents we need to check to obtain all this information. The analysis must conclude which elemental impurities need to be included in the risk assessment and their concentration on each source of contamination. Once we have gathered all this data, we can move on to the evaluation itself. As we already mentioned, there are 4 different options for the evaluation. In option 1, it's assumed that the maximum daily intake of the product is less than 10 grams per day, establishing this limit as a conservative approach. In this option, the actual proportion of the components in the drug product is not taken into account, but it's considered that they could be present in any proportion, being 10 grams the maximum possible amount of each component. The concentration limit is calculated for each elemental impurity by dividing their PDE value by 10 grams. This limit is defined as the maximum concentration of a certain elemental impurity that could be present in each component, so the PDE would not be exceeded. The advantage of this option is that it does not require further calculations. We only need to compare the individual concentration of elemental impurities in the components against the concentration limit. However, this option is highly conservative, and then positive evaluation will rarely happen. Option 2A is similar to option 1. However, it considers the actual maximum daily intake of the product instead of 10 grams. As in option 1, the actual proportion of the components in the product is not taken into account, but it's considered that they could be present in any proportion. The concentration limit of each impurity is calculated by dividing its PDE by the maximum daily intake of the product. The advantage of this option is that it requires simple calculations. We just need to divide the PDE by the maximum daily intake. In addition, this option may be positive for products with low daily intakes. However, the disadvantage is that generally this option is not accepted in products with mineral excipients, which used to have high levels of elemental impurities. Option 2B is the most complete option to carry out the evaluation by components approach, since it takes into account both the maximum daily intake of the product and its actual qualitative composition. No concentration limit is calculated, but the amount of each impurity administered per day is calculated and compared directly with the PDE. For each elemental impurity, the predicted concentration in each component is multiplied by the maximum daily intake of that component. The total daily amount of each impurity corresponds to the sum of the contribution of all the components. This option is the most complete and representative. It permits positive evaluation when there is no data of some component and when there are minority components that present high levels of elemental impurities. However, it requires more elaborated calculations for each component and each element. Finally, option 3 is based on the determination of elemental impurities directly in the finished product. Although the evaluation is carried out by option 3, it's necessary to perform all the previous steps of the risk management process, since it allows to identify the origin of the elemental impurities present in the product. A minimum of 3 different batches of finished products should be analyzed using appropriate analytical techniques adequately validated. The number of batches to be analyzed to obtain enough representative results is determined individually based on the characteristics of the product, considering several factors such as the presence of mineral components, the use of more than one supplier of one or more components, or the high proportions of certain components. The determined concentration for each impurity in the finished product is multiplied by the maximum daily intake of the product to obtain the total daily intake of each impurity. This value is directly compared with the PDE. The advantage of this option is that it takes into account the whole process and guarantees safe levels. However, it implies high analytical cost. Finally, according to the outcome of the assessment, it's determined if it's necessary to establish a control strategy. For each impurity, it's calculated a control threshold, that is the 30% of the PDE. The control strategy is determined by the comparison of the obtained values of elemental impurities against the control threshold. If the element presents levels below the control threshold, the content of elemental impurities is considered to be adequately controlled, and therefore no additional controls are required. If the element presents levels below the PDE but above the control threshold, to ensure that the PDE is not exceeded, a control action needs to be established. Specifications can be established in the final product or in the component with the greatest contribution to the final content of the impurity in order to establish a maximum permitted concentration that allows compliance with the PDE. If the element presents levels above the PDE, it's necessary to establish control over the process. Once the cause has been identified, alternatives are proposed, such as changing the suppliers of raw materials, modifying the manufacturing process, incorporating additional steps during manufacturing, or changing the packaging material for a more appropriate one. In certain cases, a level of elemental impurities higher than the PDE may be accepted. These cases include situations in which the product is administered intermittently and for short periods of time, or in special indications in which the benefit-risk balance is positive. Each specific case must be studied to make an appropriate toxicological justification based on the specific information of the product. Regardless of which of these options is used, the workflow of the process is the same. First, all the necessary data needs to be collected, either from the drug product or from the components. If necessary, experimental analysis may be performed. In both cases, all the data is summarized and the concentration of elemental impurities is tabulated. This summary must include also the potential contribution from other sources, such as the equipment or the container closure system. Then, evaluation is performed following the chosen option, for which the concentration levels or the total daily intakes of elemental impurities are compared with the concentration limits or the PDEs respectively, depending on the followed option. Depending on the outcome of the assessment, there are 4 possible results for each elemental impurity. If the element is not intentionally added nor likely to be present according to the route of administration and the results of the analysis, the element is excluded for the risk assessment. If the element is intentionally added or likely to be present, the total daily intake is compared against the PDE and the control threshold, as I mentioned in the previous slide. Now, my colleague Sandra will explain you the following parts. Thank you all.
Thank you, Marta. Thank you very much. Sandra, it's your territory now.
Thank you, Marta. Thank you. I'm going to share my screen. Okay, I think you can see now.
Yes.
Okay.
But está con las diapositivas a la izquierda.
¿Se ve?
En modo lectura lo puedes poner.
Sí. Sorry.
Ahora.
It's okay, dear. Now
Okay, sorry.
Perfect, yes. No, it's okay.
Okay. I think, okay, now. So, thank you, Marta. Hi, all. As Jana said before, I'm Sandra Paredes, Toxicology and Quality Assurance Technician at Azierta. And I'm going to continue with the next part of the training. This part is related to the documentation that we are going to need to perform the risk assessments. So in this slide, we are going to review the different type of documentation that we are going to need depending on the component assessed in each case. First, for the finished product, we are going to need the unitary formula. Also, if there are specifications regarding the elemental impurities in the product, and if analytical data is available for this final product. In the case of the API, we need to know the number of suppliers that we have. It could be only one or more than one, and ask them regarding the elemental impurities information of their product. We have our own questionnaire available for the manufacturers to be filled, but in most cases, they have their own specific Q3D statements. Also, we need to know the origin of the API, if it's synthetic, produced by fermentation, etc., and if they use catalysts or intentionally added elements during the manufacturing process. It's very important to obtain analytical data of the elemental impurities of the API, of the elemental impurities of class 1 and 2A in all the cases. As Marta said before, we need this data in all cases, and also the data of the intentionally added elements, if there is any. In the case that the product is administered by the parenteral route or by inhalation, we are going to need also the values for the class 3 elements. Also, if specifications are available for the API, it's good to know. Regarding the excipients, as in the previous case of the API, we are going to know if there are one or more suppliers of this excipient and their elemental impurities information. Also, the origin of the excipients, if there are mineral, animal, synthetic, or whatever, and analytical data or specifications if they have some. Depending on the origin, the excipient will have different levels of risk that this we are going to see in more detail later in another slide. Sometimes for the excipients, we don't have any information from the manufacturer. And in these cases, we can use information from the literature, perform a literature review, and we can perform the risk assessment based on prior knowledge. The next one is the packaging material. The documentation that we are going to need is the composition of the materials, especially the ones that are in direct contact with the product. It would be the primary packaging, as well extractable and leachable studies of the components if they are available and only if it's applicable. Also, we can perform a review in the literature of the components of this primary packaging. Regarding the manufacturing process and equipments, we need to know first the process flow. We should know all the steps that have the process. I give you an example, for example, for a tablet, mixing, granulation, and compression. Then all the equipments that are used in this manufacturing process and the materials, as I said in the packaging, especially those that are in direct contact with the product. The most common material in the equipments in pharma is the stainless steel. Also, we need to know the conditions of the different process, if they are aggressive or not, the kinetic energy, the pH or temperature, and also if the equipments are qualified and if the GMP practice are followed. Finally, for the service, we should collect information regarding the services that are used during the manufacturing process. Typical service use are water, usually purified water or water for injection in the case of parenteral products, and sometimes there are other organic solvents as ethanol. Pure steam is used for the sterilization of the equipments and also the primary packaging, the vials in the parenteral products. And also compressed air could be used, that is typically used to transfer the product between equipments. We are going now to see how the risk assessment is performed for the previous components with all the information that we have collected. So, sorry, the services. As we saw before, the services typically used are water, steam, compressed air, etc. According to the guideline, the inclusion of elemental impurities by this source is low and can be reduced following the specification in the pharmacopoeia and following the GMP practices. It's important to remember that this section should not be taken for granted, since, for example, water can be a critical raw material. One important parameter in the water is the conductivity that is related with the presence of ions. So if we measure the conductivity routinely, the probability of detection of the inclusion of these ionic elemental impurities is high. The risk assessment of the services can be done and grouped for more than one product. For example, purified water used for the product is the same for all in one manufacturing plant, so the risk assessment could be done only once and use it for all the reports. The risk analysis is based on the materials, the in-process controls, analytical measures as the conductivity, qualification of the service, detection of corrosion, abrasion, and the maintenance and cleaning. Now we are going to see how to perform the risk assessment of the manufacturing equipment. We need to know, as I said before, all the process that has the product and the conditions. In this slide, you can see typical example of different process. We perform the risk assessment considering the kinetic energy of the process and the conditions. Then we also consider the qualification of the equipment and if its inspections for abrasion or corrosion are performed. With this information, we give an overall risk to the process. This is done with a failure and effect analysis model. For example, in the table, in the first line, we have a granulation that could be dry or wet. In the case of the wet one, the kinetic energy is high. And since it's a wet granulation, the product is wet, so the conditions are going to be aggressive. So it could exist a risk for the product to extract the elemental impurities that could be present in the material in contact with the product. However, if periodical visual inspection for abrasion or corrosion are performed in the equipments, it's easy to detect the possible extraction of these impurities, so the risk remains low. In the case of the dry granulation, the conditions are not aggressive and neither the kinetic energy, so the risk is low. You have here also other examples where the conditions of the process could be also aggressive. As you can see the mark in red and in yellow. As for example, we can have high temperatures during the manufacturing process, basic or very acid pH, but if appropriate controls are stayed, the risk remains low. For example, in the case of the particle size reduction that we have in the last line, the kinetic energy is very high and the conditions are very aggressive. So we can have a potential metal transfer to the product. But in most of the cases, the GMP routines will be enough to keep a low risk. But it's important to say that all process should be assessed in a case-by-case basis. The next one is the packaging material, in which we have, in this slide, we have a figure in which different packaging materials and different types of products are covered. We are going to start with the green box that represents the lowest risk, since it's the case when we have a packaging with low concentration of metals and a drug product with a low interaction with the packaging. This would be the example of a solid product. Then we have the other two gray boxes. That is the case of a good solubility of the metals in the drug product, but if the packaging material has a very low content of elemental impurities, the risk of inclusion of elemental impurities is low. But then we can have a limited solubility of the metals in the drug product and a limited interaction between the product and the packaging material, but the metal content in the packaging is high. So in this case, we should assess further. This could be a case of a semi-solid product, of a cream, for example. Then the red box is the highest risk, that is represented by a high concentration of metals in the packaging and a high degree of interaction between the product and the packaging. This is, for example, the liquid products. And in these two last cases, we will need to review the other data, as for example, the extractable and stability data. Finally, say that the packaging that is used during a very limited time are not expected to be at risk. For example, the inhalation devices are only in contact with the product in the moment of the inhalation, so the risk will be low. Now we are going to see how to perform the risk assessment of the components, the API and the excipients. So for the excipients, the risk assessment for each one can be performed through the source that we have seen before, the supplier's information, the literature review or analysis if they provide us the data. As you can see in the slide, the synthetic excipients without a metal catalyst are the ones that have the lowest risk. Also, the animal and plant origin excipients has a lower risk. This is the case of lactose or cellulose derivatives. Then we have the synthetic excipients with a metal catalyst that have a higher risk. This is, for example, the case of mannitol, that is usually manufactured with a nickel catalyst. And then we have the last one, the mineral excipients, as for example, the talc, that represented the highest risk of the excipients. This is because the high content of elemental impurities in the mineral products and the high variability. It's important to remark that, that when we don't have information from the manufacturer regarding the excipients, the lack of information should be covered with wide margins of safety. For these cases, in which we don't have information from the manufacturer of the excipients, Azierta has developed an excipient database with more than 500 excipients, and we are increasing this number day by day. This is an advantage in relation to the knowledge of the potential elemental impurities in each excipient. This database is fed by the suppliers every day, but it's very important to remark that this information should be always reviewed by experts with knowledge in the material, in the guideline. And finally, we have the risk assessment of the API. The approach that we have set for the excipients is not possible to be followed in this case. This is because the majority of the incoming products in the synthesis of the API are later eliminated. In the synthesis of most of the APIs, we have washing steps, extractions, purification, distillations, etc. So all these steps are going to reduce the risk of inclusion of elemental impurities in the final API. As well, the same final API could be manufactured by different synthetic routes. Therefore, the analysis of the final drug substance is the best approach for this component. It's also important to remark that the manufacturer of the API should communicate us if there are any changes in the synthesis or in the product, because this could lead to a different risk assessment, and then our evaluation and conclusion of our report could change. This is important to reflect this point in the technical agreements with them and in the audits. Now we are going to see, once we have performed the risk assessment for all the potential source of contamination in our product, we are going to see in the following slide how to present these results. The documentation of the risk assessment could be requested directly from the health authorities, but for example, in the case of a new product, the risk assessment must be documented in the regulatory file. There is no need to present the entire risk assessment. We can present only a summary of the risk assessment strategy that we used, a summary of the elemental impurities that we have identified as a potential contamination in our product, with the total levels that we have observed or the levels calculated for the product, and also the conclusion. As well as if we analyze the product or maybe one of the components of the product, we should present the data. Sometimes the health authorities could propose some controls at the time of the registration of the product. Finally, I want to talk to you about the life cycle of the risk assessments. When the risk assessment is finished, although your product is complying with the guideline, after some time, new information could be available. If this information refuses the risk assessment that we have performed before, we are going to need to perform the appropriate changes in the document and correct the risk analysis and perform. Sometimes you have to perform again if you have any new data, the evaluation and the control strategy if it's necessary. Any changes could lead us to update the control strategy stated before, if there was any, or evaluate if the one stated before is still applicable to control the elemental impurities and not exceed, in any case, the PDE. Well, so thank you so much for your attention. Now my colleague Marta is going to show you how to perform a few practical cases.
Thank you, Sandra.
Hi again. Okay. So in the following slides, we are going to show you different possible scenarios that you may find in the risk assessment. In the first case, elemental impurities are assessed in a drug product presented in capsules containing film-coated microtablets for oral route. This drug product has several strengths, from 20 milligrams to 60 milligrams. In order to obtain the maximum daily intake, we need to know the quantitative composition of each strength to identify the worst case for each component. In this case, the worst case for all of them corresponds to the 60 milligrams capsules. Taking into account that each capsule weighs 278 milligrams and that the maximum recommended posology is 2 capsules per day, the resulting maximum daily intake would be 556 milligrams. After analyzing the manufacturing process, we identified that the potential sources of elemental impurities in the drug product are purified water, manufacturing equipment, container closure system, the active substance, and excipients. Each of these sources need to be assessed to identify their contribution to the final content of elemental impurities of the drug product. As per the components, the maximum daily intake of each of them must be calculated to perform evaluation by option 2B. For this, the total weight per capsule is multiplied by the maximum recommended posology. In this case, we should multiply these values by 2, and then the obtained values should be converted to grams. In the following step, evaluation by options 1 and 2A is performed. Remember that these options compare the concentration of each element in each individual component against a concentration limit. These values are obtained using the statements on elemental impurities provided by the manufacturer of each component. All of them are expressed as micrograms of the impurity per gram of the product, or in other words, in ppms. You can see that some of them provide white margins, for example, for the active substance, for triacyl citrate, and for the capsule, their manufacturers declare values of 10 and 30 ppms based on the heavy metals test. For other components, for example, hydroxypropyl methyl cellulose or talc, the results are based in analytical data and hence are considered more representative. If we take a look to the outcome of both evaluations, we can see that option 1 is not accepted for any element, since the concentration on one or more components exceed the concentration limit. In the same way, option 2A was not accepted for lead and cadmium. To obtain more representative results, evaluation by option 2B is performed too. In this case, we don't compare concentration, but total daily intakes of each element. For this, we multiply the concentration of each elemental impurity on each component by its maximum daily intake in grams. The obtained values are summed to obtain the total daily intake in micrograms per day, which is compared directly with the PDE. We can see that the total daily intake of lead and cadmium is above the PDE due to the contribution from the active substance and the capsule. Taking into account these results, we are going to raise 4 different options and then we will explain which one is the correct. Perform experimental analysis of lead and cadmium in 3 batches of drug product. Perform experimental analysis of lead and cadmium in 3 batches of active substance and 3 batches of capsules, which were the major contribution to the final content of lead and cadmium. Request further information to suppliers of active substance and capsules, or perform a toxicological justification for exceeding PDE of lead and cadmium. In this case, we know that the concentration of elemental impurities on the active substance and the capsules is overestimated, since the manufacturers have provided large margins of 10 and 30 ppms based on the heavy metals test. As we mentioned at the beginning of the training, this test is very unspecific and the sensitivity is very low. Therefore, it provides detection limits too wide. The actual concentration of elemental impurities in these components is expected to be far below these values if we use adequately validated analytical methods. For this, the most suitable option would be to request further information to the suppliers of the active substance and the capsules. Let's assume that we can get a new statement for the capsules. Taking into account the new information provided by the manufacturer of the capsules, we perform a new evaluation by option 2B. In this case, the concentration of elemental impurities in the capsules was measured using ICP-MS with a limit of detection of 0.5 ppm. The obtained results demonstrate that no elemental impurity was detected above this limit. With this new information, the evaluation by option 2B is accepted for all the elements. However, when we compare the total daily intake with the control threshold, we can see that the levels of lead and cadmium are above this value due to the contribution of the active substance, for which we are still using the values obtained using heavy metals test. Therefore, we could now consider the following options. Set a specification for lead and cadmium in finished product, request further information to active substance supplier, perform experimental analysis of lead and cadmium in 3 batches of active substance, or it's not necessary to do further actions. As we have already mentioned, the concentration of elemental impurities in the active substance is overestimated with the heavy metals test, and for this it would be necessary to request further information to the active substance supplier. If it's not possible to obtain a new statement with more accurate data, we could analyze lead and cadmium in 3 batches of the active substance to obtain more representative results, and then update the evaluation by option 2B. In the next case, we are going to assess the content on elemental impurities in a drug product presented in oral effervescent tablets. The maximum daily intake of this product is 17 grams per day, since according to recommended posology, 5 tablets per day may be administered. As we did before, all the potential sources of elemental impurities need to be identified and assessed. In addition, the maximum daily intake of each component needs to be calculated according to the qualitative composition of the product. In this case, evaluation by option 1 cannot be performed, since this option is only valid for those products with maximum daily intakes below 10 grams per day. As per option 2A, evaluation was performed using the information provided by the manufacturers of each component. You can see that the manufacturer of trisodium citrate has declared that all the elements present levels below 0.5 ppm based on analytical results. On the other hand, the manufacturers of potassium bicarbonate and lactose monohydrate declared that their products have an internal specification on 3 ppm and 5 ppm respectively for all the elements. Taking into account all the available information, evaluation by option 2A cannot be accepted for any element, since the concentration limit is exceeded for all of them in 1 or more components. When we use this data to perform evaluation by option 2B, we note that all class 1 elements exceed the PDE values due to the main contribution of trisodium citrate, potassium bicarbonate, citric acid, and lactose monohydrate. Therefore, now we could raise 4 options. Perform experimental analysis of impurities in 3 batches of drug product. Perform experimental analysis of impurities in representative batches of each component with major contribution. Request further information to suppliers of raw material, or perform a toxicological justification for exceeding PDE of arsenic, lead, mercury, and cadmium. Although we know that the concentration is probably overestimated in the components with major contribution, taking into account that several components are involved, it would be more difficult to obtain new statements from all the manufacturers. In addition, if we decide to analyze 3 batches of each component, it would require high cost. As per the toxicological justification, it's the last resource that we can use when it's proved that some element presents toxic levels using accurate information. For this, in this case, the correct option would be to perform experimental analysis on 3 batches of the product. The analytical method must be validated for a screening test, and the limit of detection should be low enough to guarantee that the obtained results are enough representative, and permit to demonstrate compliance with the control threshold considering the maximum daily intake of the product. Now, Sandra, we will explain you the next case studies. Thank you all for your attention.
Thank you, Marta. You can see now my screen?
Yep.
Okay, so I'm going to present you 2 more practical cases. And the first one are tablets. Okay, now, so here we have the case presentation. Here are tablets that are administered orally, and we have different tablet strengths. The dosing schedule is 1 tablet maximum per day. So if we multiply the weight of each tablet per the dosing schedule, that is 1, we see that the maximum daily product weight, in this case, the worst case, is for the 60 milligrams tablets. In this slide, we have the composition and identification of all the source, all the quantitative and qualitative components, the API and all the excipients of the product, and then the potential source of contamination, in this case, water, manufacturing equipment, the container, the APIs, and the excipients. Here you have the evaluation by options 1 and 2A performed, and as you can see, the evaluation by option 1 was not accepted, and this is mainly because the data of the API, that is 20 for each elemental impurity, and also the evaluation by option 2A is accepted for some of them, but for lead, cadmium, and lithium is not accepted. As you can see, we have levels, if you compare, you have levels in the API that of 20 that is higher than 8 in the case of lead and cadmium. This is why this option could not be accepted. Then here we have the evaluation performed by option 2B. And as you can see, all the elemental impurities were accepted by this option. But regarding the control threshold, all the levels were below the PDE, but in the case of lead and cadmium, the levels were below the PDE, but above the control threshold of the 30% of the PDE. As you can see marked in red, this is mainly because of the data of the API and also the lactose that contributes a lot, and the total levels of lead and cadmium is higher than 1.5. So an action should be implemented. As a conclusion of this case, this product complies with the option 2B, but requires an action for the elemental impurities, lead and cadmium. The risk assessment demonstrates that the focus is placed, as we saw before, marked in red, on the API and the lactose. It's decided to wait to have better information from these components from the suppliers, And once we receive the information of the manufacturer, for example, of the lactose, we update the risk assessment, the evaluation. So here you have the new table when we receive this new information from the lactose. And as you can see, now the levels of the lactose contribute much less. So now the control, the outcome is no control for all the elemental impurities because they are below the control threshold. So when should we update this risk assessment? We have here 4 options. The first one, the risk assessment will require an update with any change in the manufacturing process, raw materials, etc. It does not require any update because now with the new data of the lactose, it complies with the limits. It could be updated if the manufacturers of the API or excipients provide further information. And the last one, it could be periodically updated in a yearly basis. So the correct answer is the first one and the third one. Is so important to remark that with any change in the manufacturing process or our raw materials as the API or the excipients or the packaging, if we have any change, we should evaluate again the risk assessment. And also the third one, because if the manufacturer of the API or some excipients provide further information, we should also analyze them. So now the last case that are for vitamin B12 tablets. So in this case, the tablets are administered orally also, and we have the product weight, but in this case, the tablets, the maximum dosing schedule is 2 tablets per day. So if we multiply the product weight by the maximum dosing schedule, we obtain the maximum daily product intake, that in this case is 0.782 grams. And the clinical indication of these tablets is the vitamin B12 deficiency. So now we have here the case presentation in which we have all the excipients of the tablet and also the API, that is the cyanocobalamin. And also the potential source, as always, of contamination. In this case was purified water and isopropyl alcohol and all the rest of potential source that we have seen before. So in this case, an analysis of the final product by the evaluation of the option 3 was performed at first. So here you have the results of the analysis. As you can see, marked in red, the results for cobalt are very high, are 143.3 ppm. So this is so high, and we should know where this comes from. So the options of this case could be the first one, we can perform a toxicological justification for exceeding the PDE of cobalt. A validation of analytical method to determine the quantity of cobalt in the finished drug product. Set a specification for cobalt in the finished drug product and only release the batch if the levels of cobalt does not exceed the PDE. And the last one, the risk assessment, perform a risk assessment to determine the source of cobalt. So here the correct answer is the last one. Although we analyze the final drug product, we always need to perform first a risk assessment to determine where the potential source of contamination of the product. So especially in this case, that I remind that was a vitamin B12 tablets, cobalt is part of the structure of the cyanocobalamin API. So this will be one of the out of scope cases that Marta said at the beginning of the presentation, because cobalt is part of the molecule of the API and it's needed to the therapeutic effect. So in this case, the cobalt will be out of scope and we don't have to assess further. So that's all. Here you have also our services at Azierta in the toxicological department, as you know, the PD reports, nitrosamines evaluation, and of course the Q3D elemental impurities evaluation. So now I think we can go through the questions part.
I will try to help you there, dear. I will tell the question and you will see how you can answer it. The first question we have from Mr. Akram is the manufacturer supplied the material with certificate of analysis claims that it is free from elemental impurities. Is it enough to skip the test?
Nope.
Well, it depends. It depends if, sorry.
It's okay, go ahead, dear, sorry.
Maybe I can answer and then Marta can complete my answer. I think it depends if the API, we are going to need the analytical data from the manufacturer, but as I said, for example, if it's an excipient that is not mineral, we can perform a literature review and also from the data from other manufacturers of these excipients, and with safety margins, we can perform the risk assessment. But in the case of the API, we need the analytical data. I don't know if Marta wants to add something.
Go ahead, Marta.
Generally, in the certificate of analysis, the manufacturers does not provide a lot of information about Q3D. For us, it is best to obtain the elemental impurities statement that each manufacturer should have his own. If they have not one, you can prepare a questionnaire asking him, for example, if they use intentionally added elements, or asking if they have measured the levels on at least 3 batches, or if they have performed a risk assessment. But usually in the certificate of analysis, they don't include a lot of information about Q3D.
Yes. I totally agree with Marta, and I think you should do some sort of research alone, because just a statement or يعني, how do I say it, commitment from the
Yes.
supplier is not enough. It needs to be verified. يعني, it needs to be proved with the studies.
Exactly.
Okay?
Yeah.
The second question is why RA is mandatory upon following option 3 for evaluation, testing of drug products. This comes from Ali Abu Awad. I think why should we tell the authorities about option 3 for evaluation?
It's for our experience, authorities always ask for the risk assessment. If you only analyze the product and perform the evaluation by option 3, they probably will send you a letter with observation that you need to identify all the potential sources and evaluate the contribution, the risk. Maybe if you don't have information about the component, at least you should perform an assessment, for example, based on the origin or the, I don't know, the use of metal catalysts to identify which components have higher risk and which one have lower risk. But only submitting the report with the analysis, it's not enough for the authorities. They always ask for everything.
Yeah. The second question third question is from Fadi. The results can be taken from supplier certificate of analysis, or to do we have to do our own analysis. We have answered this question. We have to do the same analysis.
Yep.
Hassan, can we depend on analysis of heavy metal from certificate of analysis supplier only? The same question again. No, you can't. I have a question from Mustafa Mahmoud. Hi. How usually we can control or evaluate the elemental impurities in compressed air?
Well, so generally, as the guideline says, the risk of inclusion in compressed air and in all the services is low. Specifically in the compressed air, the elemental impurities could be only present in the air in particles. So if you control the air of your plant with filters, and for example, if your air is free of oil particles,
Yeah.
I think with this it should be controlled.
Yeah, I think if you have a validated system for your compressed air and you do يعني routine monitoring and routine evaluation for your filters and your system as well, I don't think you need to do any further testing. That's what you mean, ladies. Yes?
Yes.
Yes.
Sorry, I would like to
Yes.
say something about the previous question. When someone asked if the heavy metal analysis on the COA is valid, the heavy metals test is obsolete. So if they send you a certificate of analysis, for example, with 10 ppms for lead, it's not valid. You need to request information. Okay.
Yeah. Yeah, the heavy metal test is obsolete. We have to rely on risk assessment related to elemental impurities.
Yes.
Okay. Fadi, again, the analysis should be done for each batch.
No, I don't know if it's for the final product or the API.
Final product, finished product. Here in the region, most of the manufacturers are manufacturers of finished products.
Okay, so the guideline says that you should analyze 3 batches. But for example, if the variability between the data of these 3 batches is high, sometimes you need to analyze more. But generally, with 3 batches is enough.
Okay. And if we're talking about raw material, API?
Yeah, the same.
The same. Okay. Ali Abu Awad, he has another question. To cover all sources of elemental impurities, is it recommended to test the drug product before packaging, after packaging, or after stability study? If after stability study, what STP conditions and which interval?
It depends on the outcome of the risk assessment. Here you have why it's important to perform a risk assessment before analyzing the finished product. Because, for example, if you don't identify a risk from the container closure system, you don't need to analyze it before, after the storage. Okay, so for example, if you have a polyethylene bottle, it does not contain elemental impurities, or the manufacturer has sent you an elemental impurities statement on which they declare that they have performed a leachables study or an extractables study, you don't have a need to analyze the product after packaging. Okay, so however, if you are using some special packaging that you think it may pose a risk of inclusion of elemental impurities, maybe you could analyze it before the packaging at the release, and then you could analyze, for example, after 3 months and after 6 months, but it's not defined. It depends on your assessment.
Yeah, you design the interval by yourself depending on the risks that you see in the process.
Yes.
Or in the packaging, on the materials. Okay. I have another question from Ahmed Bahari. Is the plastic tube in the packaging material represent less risk on the product than aluminum tube in the concept of impurities?
No, it depends on the product, because it's, as I said in the presentation, it's not the same to have a solid product or a liquid product. The interaction between a solid product and the packaging is very low. So even you have elemental impurities in the packaging, if the interaction is very low, the risk of inclusion of these elemental impurities in your product is low. But then if your product is not solid, then you have to assess extractable data or some studies in the literature to know if your product could extract in different, for example, temperatures or different parameters, if your product could extract these elemental impurities from your packaging. But with our experience, plastic and aluminum packaging are don't represent a risk.
Okay. Okay. I have another question. Can we have the slides, please? With the permission of Azierta, if you need the slides, please contact us at Agon, and we will send you the slides. Another thing, if you need any support in the elemental impurities, contact me personally, and I will try to connect you with Azierta to make sure that you can see all the range of services regarding the elemental impurities. If you can't do this yourself, contact us. We can help you, us and Azierta, in doing this. Is it mandatory to perform the test to comply with the Saudi FDA requirements? Ahmad Asim. The test is already implemented in Europe, and I think the Saudi FDA is going through the fixed certification, which means that they will enforce the test soon. I have been talking to several authorities in the region. One of them is the Saudi, and I think this is one of the top subjects that they are talking about now. I'm not sure if they have issued the official paper to make sure that everybody is taking this into consideration. I know that the Jordanian has done that. Jordan FDA has done that. But I will see. I will check, Ahmed, and I will get back to you. Okay, because I don't think the ladies, they have the answer to this question. this is for me. I will check, Ahmed, definitely, and get back to you. Again, Ahmed Asim, how to perform the risk assessment for the primary packaging?
Well, I think we more or less answered this question before. It's all taking into account the packaging material, if it has elemental impurities in the composition or not, and also taking into account the interaction between the packaging material and your product. It is solid or semi-solid, as a cream, or it represents a higher risk if it's liquid than if it's solid.
Yes.
Okay.
I don't know if Marta wants to add something.
I think that's it. Maybe asking them for the statement to the suppliers, or if they have some documents to demonstrate compliance with some regulation, like food contact regulation, which limits the presence of heavy metals in the packaging materials. Literature review with this.
Okay. Okay. I have a question. I think everybody has this question in their mind. What if my methods for the analysis of these elements are not sensitive enough?
With the most common is ICP-MS, inductively coupled plasma mass spectrometry. It's the most sensitive, and it allows to use low limits of detection, and usually is this method the choice.
Yeah, this is the method of choice, guys. So if you don't have the equipment at your facility, you need to contract this service from outside. You need to see who is
Yes.
providing, who's the lab that is accredited and providing these services, and they should be. You should make a contract with them, and they should be able to test these elements for you. If you don't
Yes.
have it at place, okay. I have. What if the steam does not come in contact with product? It is used for terminal sterilization only of closed container and SIP of machines. So What if I'm using steam, pure steam for SIP sterilization, and do I have to do the elemental impurity for that steam? Although it's not in direct contact. Yani, I wouldn't agree, Doctora, that it's not in direct contact, because if we are talking about sterilizing steam, it's in direct contact, because at the end it will remain on the machine and the product will come onto that machine. But I will leave the answer to the ladies.
Yeah, I agree with you. First, if it's not in direct contact with the product, you don't need to include this assessment in the report. Yeah, but if it's in contact, yes, you have to include it.
Yeah, and I think this is in direct contact with the product, because the machine, it stays on the machine. This water condensate, it will stay on the machine. I have Dr. Amjad Abdel Haq. In the risk assessment, is it generally acceptable to depend on analytical data from ICP-MS quick screening, or a method should be validated before conducting any measurement?
Well, when you want to obtain information to perform the risk assessment, like informative information, it only needs to be validated for specificity and detectability. It's not necessary to be fully validated. However, if you need to monitor the presence, for example, if you detect that lead is above the control threshold and you need to monitor the levels in all the batches you are producing, this method to detect this element should be fully validated for all the parameters. But for a screening test, only for information, it's enough with detectability.
Okay.
Yes.
Yeah, great. Dr. Riham again, how often to test finished product for elemental impurities? Should we purchase ICP for routine testing?
No. As I think I said before, if you analyze 3 batches of your product and all of them are in compliance, the evaluation is by option 3 is correct, and you don't detect anything. You don't have to analyze your product unless you have some changes in your product, as for example, if you change the API or you change anything. But if you don't change anything, it's enough to analyze 3 batches.
Yes.
Okay.
Most laboratories perform the analysis with an external laboratory.
Yeah.
I think none of them has an ICP.
Yeah, I know this. I don't know, Dr. Riham, if she is in Jordan or in Saudi Arabia or in Egypt, but I know in Jordan we have a lab that helps companies in such cases. And in Saudi Arabia we don't have. Or at least there is something is going on now, but there is nothing solid now. In Egypt they have quite number of laboratories that they can contract. Anas Qasim, أهلا يا Anas. According to experimental analysis of IMP in 3 batches of DF, where can I find the reference? What is the reference for that?
The reference?
He has a question on the chat box. He says, according to experimental analysis of IMP in 3 batches of DF. I don't know what are the abbreviations.
Finished product, no?
Maybe finished product, I don't know. Where I can find the reference.
I don't know what it means.
And I can't understand the question. So, Anas, if you're still there, please elaborate your question. If you're not there, I will call you tomorrow to elaborate the question and see what do you mean by it. Okay. Anas, again, IMP, impurities, and...
Yeah, but I don't understand what with reference.
Dosage form. 3 batches of the dosage form, where I can't find the reference. He wants to know what is the reference to take only 3 batches.
It's on Q3.
Okay.
Q3D.
Thank you, Dr. Riham.
Thank you, yeah.
On Q3D guideline, it's stated that with 3 industry scale batches is enough. And if it's a pilot scale batches, it would be necessary to analyze 6. However, it depends on the characteristics of your product. For example, if you have a lot of mineral excipients in this product, which gives a lot of variability in the elemental impurity content, maybe you should analyze more batches than 3. Or for example, if you have different suppliers of API, for example, imagine that you have 2 suppliers of your API, you could analyze 2 batches using one supplier and 2 batches using the other one supplier. You have to,
Okay.
to check the information of your product.
Okay. I think this is all the questions that I have. I have another question in the questions and answers. They are asking questions now. This is the last question we're going to take, guys. Does the use of disinfectants in sterile zones considered a source of elemental that deserves to be studied?
No, I think no,
And
because
detergents in cleaning.
no, because they are not in direct contact with the product.
Okay, because they are not in direct contact, I have the same answer, but this has to be covered as well in the cleaning validation, because
Yes.
the limit of the detergent remained on the machine. It should be verified to make sure that I don't have a limit that exceeds the ADE values. That would mean that I am in the safe side. I think we have one thing in the chat box. Thank you. That's a thank you. Thank you, Ilak, ya Mustafa. Thank you, everybody from Azierta, Sandra, Marta, Leticia, Eduardo, Pablo, everybody, Danny. Thank you very much. Thank you for all the attendees. Thank you for staying with us this late and for participating in this webinar. It was very, very informative. For me, it was new information, all of them. So thank you very much. I really enjoyed it. See you next week, everybody. Next week, we don't have the subject yet, but it's gonna be probably me. I'm gonna do a webinar. See you next week, next Wednesday. Thank you very much, and see you all soon. Thank you, guys from Spain. Stay safe. Stay at home.
Thank you.
Thank you.
Oh,
Bye-bye.
bye.
Bye-bye.
Bye.
Bye.
Bye. Thank you.